So let's see if this remote's working. You're setting me up before. There, it does. So what I do before lunch is I want to walk you through something that I've been learning. It's this thing called phenomics. Now, the name phenomics comes from 1996, almost 30 years ago, and it was really looking at plants. And the idea was, hey, can we start to understand the biology of plants, look at the phenotype, basically what plants exhibit, and basically do some measurements of the soil and whatnot. It was picked up by this gentleman who's become my mentor, Leroy Hood. He's an Erasmus fellow. I don't know if you guys know Lee, but he's sort of one of the original inventors of the original sequencer. And the idea in that also Amgen, and he basically did the Human Genome Project. And what I'll do today is I want to walk you through this thing of phenomics, and I'll show you basically these mental models that were being used and pervasive throughout this, and you'll see some of that in the talks you saw earlier today. So there's this phrase, you now see it a lot in venture capital, on the lips of venture capitalists. You see it in Boston. It's in silico. You're all doing much of this yourselves. And the idea is, it's kind of a simple idea. Can we find drugs? Can we detect what's happening in our bodies in computers? It's a lot cheaper and faster than doing it in a lab. And what Lee and others are thinking about is this spinning on the left, which is you've got some biology up here, and you sample it with some sort of technical technique, basically next generation sequencing, looking at mass spec, and that gives you some basic data that you apply to technology. And that sort of has a digital twin. You say, I think this might be happening. And so what NASA treats that as, they call that the idiot savant. So for example, in the NASA version of this, the data is looking at Mars, and if you want to land, that goes into an AI, and the AI says, I think this is a good landing zone. Then in the computation, it then uses old school machine learning to say, yep, it's a good landing zone, actually lands the lander. That actually happened. So they don't really trust AI, and they don't either in drug discovery, but they use this to basically do a computation and then go test it in the lab. That gives you better technology, and you keep spinning the wheel. That's sort of the idea behind phenomics. And what you see on the right is sort of the things they're looking at in these correlation graphs. These are pretty things you can draw, where you basically take the top 100 or so different elements, you draw a line between them in terms of if they correlate. The thicker the line, the higher the correlation. So looking at things like genomics, your microbiome, clinical labs, and whatnot. And that's basically the idea of doing, can you do stuff in silico and use it to complement in biology? It's not separate at all. And the breakthrough really comes from, this is a Nobel Prize from October this year. That's how new this is. In October, they have artificial atoms called quantum dots. And I'm reminded of a lecture from 1969. They said there's plenty of room at the bottom. And what they're thinking are to see things at a nanoscale. Now, a nanoscale, to give you an idea, if the paper that may have cut your hand, that's 100,000 nanometers wide. 1 millimeter, that's 100,000 of those things. If you take a cubic millimeter, that's an exabit of information. So to give you a sense of an exabyte is all the information on the internet in 2012. Will fit in a cubic millimeter at a nanoscale. And, well, can we now start to look at our bodies and chemistry at a nanoscale on a quantum level and understand what's going on? And there's a new friend of mine, Jennifer Dione at Stanford Lab. You know, I was thinking about this, and when Lee started the Human Genome, it was about $1 billion to read the data. It's about $5,000, $6,000 a few years ago. And she's working on a technology that's about $1. And I go, how can you read this? She says, well, what I can do is, and that's her hand holding up a little slide here. And what she's doing is she has a new sensor that uses nanophotonics, which means at a nanoscale, what ends up happening is light starts to have electromagnetic properties. And then what that means is the molecules themselves have a signature, and she can actually read the signature on a single cell. And I said, that's pretty cool. How many can you do? Like one per slide? She goes, no, I can do 100,000. So she can read 100,000 unique molecules, which is about enough in a typical GWAS study. And it takes about, the chip itself is about a penny. It's about $1 to $5 for the reagents. And that's now running the lab. So at Google, we looked at this in about 2010, 2012, when we realized we don't have enough compute power if we wanted to use deep learning across all of what we offered. We thought we needed 3 or 4 more Googles. And so when that happened, that was not exactly the best information for the board of directors. They said, we need a better way to compute. And so Jeff Dean and some others said, well, let's throw everything out, like everything. And so what do you mean everything? So, well, we're going to do stuff on the left. Now, on the left, this was pretty modern a decade ago. What you see are 4 stable states of an electron as it moves around a hydrogen atom. There's 4 stable states. They're superposed, superposition. And so what end up happening is the superposition is 2 sides of the same fraction between 0 and 1, 0.2 and 0.8. That's your 0 and 1. So think of now, if you're reasoning in mathematics for a new type of computing, it's no longer lopping off the signal like we did for the silicon for many years in zeros and ones. You now deal with quantum state as your core representation. It's fuzzy now, not 0 and 1. If that's your 0 and 1, well, how are you going to reason? And the silly observation was, well, there's at that time, about 7 billion, now 8 billion of us, walking around with examples of how we reason in our heads called the neocortex, which seemed like almost a laughable idea. You're going to automate the neocortex? This is 2010. And we started. And lo and behold, this thing on the right, you can see on my website, there is another observation made that, well, how are we thinking? If you look inside our heads, and there's 1,000 Brains Theory, it's a great book. You might want to take a gander at that book. If you look inside our head, there's no light inside your brain. There's no feeling inside your brain. Instead, you've got electromagnetic, you got electro charge, and you got chemical charge. And essentially, that is sort of reasoning across all these different connected neurons, and that collective charge is actually allowing you to think. And someone said, well, what if I were to take all the neurons in your body, in your brain, write them on a spreadsheet? And then what I'm going to do is I'll say, if it's off, it's a 0. If it's on, it's a 1. And you got 100 billion rows in this spreadsheet. And that's one snapshot in time. And you do it again. That is a one long tensor, was the idea. And if you look at the math behind things for activations, you actually slide it over a little bit and you scale it down. So now the center is not 0.5, center is at 0. And now you've got things between minus sigma and plus sigma. And that's kind of a Gaussian at 0. That's a tensor. And then the idea is, well, essentially, if that's what's happening in our brain, are we essentially a tensor processor where I'm hearing your voice, I see a picture of a bunny, I'm looking at some science, I talk to customers, I'm essentially doing tensors in, tensors out. On the right is from, I did this about a couple months ago, this is using Midjourney, and I said, give me a vision of what the tensors are looking like as you're producing an image, and to produce that. Kind of an interesting little side effect. Well, this idea, this was around 2011. A few years later, Chris Mason and some others were trying to figure out these 2 gentlemen. They're twins. It's Mark on the left in green, Scott on the right in blue. The twin astronauts. Now, what happened is they sent Scott up into the ISS for over 350 days, and they wanted to understand, could we actually throw everything at these 2 astronauts to understand what's happening between on Earth and in space? And so a lot of you are already doing this. I saw some talks earlier. Let's look at biochemical data, cognition data, epigenomics, gene expression, immune, metabolomics, all this. Take all these modalities, and let's just do correlations and find stuff. What's actually happening? And they call it integrative analysis, which are the pipelines you're all doing. This is 2015. And what they found, lo and behold, is Scott, who went up, the telomeres at the end of the DNA, they basically started to grow while he was in space, protecting against the invasion of all the particles that happen because you don't have the Earth to protect you. Pretty amazing. So the telomeres of Scott actually grew longer to protect his DNA from diffusion. I'll get to that in a moment. This was the first, and around this time, you've seen this before, we started to figure out a way, and this is from Alex and everything else, to say, well, what if you took all of your tensors and put them into a grid? And then what I did is I literally drew a smaller grid, and I would connect one line from the bottom to the next. And then I drew a smaller grid, and I kept doing this. And if you get to the very top, basically what I'm doing is I'm going to fake a neuron in the sense that when I connect one number to another, That's like a connection. What I'm going to do is I'll say, how good is that connection? That's a multiplier. And as it shifted, it's called a bias. So I multiply and I add. And I literally go to the very top. And this is about, and at the time, about 83 million or so of these things. And the very top is a strip. And the strip is 1,000 animals. And what you're trying to say is, if I fill it with white noise and I throw an image at it, what's it tell me is the most likely animal that I'm seeing? You say, that's not going to work. Well, it turns out when you first kick it through, it goes, no, it's not. It's a dog. So what you do is you go back and you literally chained ever so slightly the gradients so that the weights and the bias shift a little more towards dog. Do it a few thousand times, a few million times, it gets it right. And this simple idea, one of the engineers who did this, he was hired by a little company called Grail a couple years later. He went basically out and said, what if I could do instead of cat or not, cancer or not? And the idea is, can I look at not just, he said, well, cancer or not, let me go look and I'm going to dumpster dive in the blood. What? Yeah, I'm going to dumpster dive, because whenever you have cancer, it's throwing off trash, literally, and your blood's cleaning it up. That trash is called methylated DNA. Methylation opens up the DNA to replicate. And it's like a snake skin floating in your blood. You can pick these things up, and the first model was cancer or not, and it worked. That's pretty cool. What else can you do? Well, how about which organ? So take the cancer or not and then put in the organ and train it. Can you detect the organ? Yes, you can. Well, how about the stage? Oh, that's not going to work. They put in the stage and the stage works. So this is just using the CNN, a simple idea from the astronaut study, an AlexNet. Now actually is worth about $8 billion. That's one example of using a CNN. While that was happening, the idea was, well, that's detecting one word. How about language? That's really hard. Because what you want, imagine if you're trying to predict and doing a language translation, how do you then go predict each word as it goes along? If you're trying to predict language, the problem is the words change the more you see. And you can't afford a CNN for every single word. So what you needed is a programmable CNN, where the data itself affects the weights you use. You follow? So what you want is that you imagine having an infinite number of CNNs where the data itself tells you which one to use. So you learn these dictionaries. And that's the thing on the left called the transformer. The idea was, can I make a CNN so that as the data comes in, it looks in a dictionary and says, use these weights. And that's essentially the lookup dictionary of the transformer called the attention network. And that's an example of drawing attention between the most correlated bits of an image. And that was 2017. And at the same time, physicists said, I have another idea for another sequence. I'm going to take an image like this, and I'm going to add some white noise to it. It's Gaussian noise. And I'm going to do a little bit of noise and do the next noise. And the Gaussian noise basically says, give me noise around this median and this standard deviation. It's a nice little bell curve. If you add the noise over 1,000 steps, you get pure white noise. But can I relearn those different steps and play it backwards? It's called diffusion networks. And these all happened at the same time, and they hit in 2022. And along the time that hit, there's something called GenSLM. I don't know if the GenSLM people are here. This is from NVIDIA and also at a university in Germany. They put both of these together. And what they did first is they used basically the transformer to say, can I learn a lower dimensional representation of COVID-19? So take 10,000 different individual, basically the nucleotides, and learn a smaller embedding to basically represent what that is. And then can I then figure out a diffusion way to go from that to figure out the next variant? And from that, can I classify, is this a variant of interest or not, from the millions of things being tracked? And that used an awful lot of compute power, about 6,000 different NVIDIA chips, and actually won the Gordon Bell Prize, which is the Nobel Prize for supercomputing, basically using those two techniques to figure out, is this COVID actually going to affect humans or not? And then it gets better. Other people said, well, wait a second, what else can I encode as a language? They said, well, how about the sequence of angles in a protein? Because as you march along each different nucleotide, each different amino acid, you can basically talk about the proteins and the angles as they shift. That's a sequence of angles. Then you can take that and you can actually embed those things as well as images and add long-term memory to these models. And this was like about 3 or 4 months ago. There's the idea of, well, I've got the large language models, which are transformers plus diffusion, but they said, well, it's not as good as a computer. It's not as good as Google search. It's not as good as a lab tech. And these teams said, no, don't do that. Don't force the LLM to be Turing complete. Give it a calculator. Don't force it to be Google search. Let it search. And so the idea here is, if you have a task, can that go into, and your control plane now becomes the foundational model, and the language of things you do are the steps you do in the lab, the steps you do in the drug discovery, the steps you do for, and that produces different tools, and the LLM now acts like the control plane for everything else. And so this was adding tools. And that's one LLM. Well, the good old-fashioned AI crowd that I grew up with, called good old-fashioned AI, GoFi. This is the crowd that didn't believe in neural networks, and I was one of them many years ago at MIT. And they said, well, what about us? Have you heard of A-star search? A-star search that played chess. It was pretty amazing. And they said, well, wait a second. Let's make the cost function, in other words, how far I'm away from the goal, make that an LLM. Let's make the actions you can take an LLM action, and let's make the state of the world a tensor space. When you do this, you take the old school AI, and now one LLM with the ability to do tools, with the ability to represent the world in multimodal, that is a single element now wrapped into a much larger space using good old-fashioned AI. So instead of basically Lisp or symbols, you're now actually using an LLM. What do you do with this? This is pretty amazing work. This is from McMaster's University, started at MIT. And the idea was, if you go into a hospital, these superbugs are pretty awful. It's an antibiotic that resists all our treatment. It's because you clean, patient comes in, you clean it, you do everything you can, some molecules survive. And those molecules are resistant to all of our treatments. And the idea was, I wonder if there's another existing medicine. Could I use an existing medicine for something else and repurpose it as an antibiotic? Seems like a good idea, but if a medicine's $150,000 to try, it's intractable, 6,000 medicines. So what he did is he took the graph structure, you saw graph neural networks before, and you can encode this as basically a neural network. And the neural network, you can then train that network to basically predict biological properties. Basically, does this affect E. coli or not in the Petri dish? Instead of cancer or not, does this kill E. coli or not? And in 48 minutes, they basically looked through all the 6,000 drugs, and it spit out 9 different candidates. 7 of those are very quickly thrown out because they wouldn't last in the human body. They've had experiments with them. But 2 were interesting. And now they're entering FDA trials in a brand new startup. So imagine this is the idiot savant saying, I know there's 6,000, try these 9. Scientists threw out 7, now they're actually going through 2 trials, and they're using it to basically find a brand new bug. Not 7 years, but less than an hour. Now the FDA is seeing this, and the FDA is jumping on board. Some of you are probably involved in this work. They're now looking at ways, can we actually have precision drugs so that I can build a drug for you, especially for cancer? And they call these platform drugs. And I love this. This on the left is a gross simplification of mRNA-based drugs. So what you see here is that UTR is called an untranslated region. It's a piece of mRNA. And green is the actual code that your cell reads to replicate. What you actually do is you take the green piece out of a spike protein from COVID-19, you put that into this cartridge, which is the blues on either side, has a little tail, and you wrap it up in a little ball. It's about 10 nanometers wide. Those balls act like a salt and basically go into your arm, and then it goes into your armpit, and your armpit becomes a bioreactor. Your cells in your armpit read this and produce the same antigens that are on the top, and your immune system attacks. Pretty cool. That's called an mRNA platform. The one on the right is basically monoclonal antibodies, and it looks like a Y, where it's got a heavy chain and 2 light chains, and it's like a DeWalt screwdriver. It's different tool tips. And those tool tips are designed to go after certain different molecules. And so the idea is, can you improve the platform of the process? So you just basically do a code swap for the green or the tooltip, and that's how you get a personalized vaccine. When I saw this, I started having tunnel vision because many years ago, this is my mother, she passed away. That's me a few years ago. And I think she's 23 here. And my mom, she passed away, she had HER2 positive cancer. And it took her life about 20 years ago. And she said, could you somehow help people not have to go through this again? And I'm a programmer, I didn't know what to do. My brother became a radio oncologist, we're partnering together. But I really wanted to help people do this. And it turns out there's now drug trials happening last October where they're actually taking patients like my mother and creating personalized vaccines in less than 30 days and curing them in the sense that their immune system is attacking it and it's going away like a scab. And one of the patients didn't work with, and they basically combined it with another one called a PD-1 inhibitor. They used that to basically combine with another drug, and she was also cured or managed, much like diabetes. So I began my journey at Google, said, well, if we can understand all this stuff for the language in Grail, could we start to understand the language of our bodies itself, the language of nature itself? Can you start to understand what all these ATCGs and U's actually mean, was the question I asked at Google Cloud. And my brother said, oh, you mean like the Oncotype score? I'm like, the Onco what? And he says, there's something called Exact Sciences. And you can see online, you can see the actual publication of the research paper. It's a polygenic score. It's a polygenic risk score. They found it, basically look at all these different genes that affect breast cancer, and they basically created this score to detect what level or what's the most likelihood you're going to have, a metastasized cancer, and that produces a score, and that basically drives an entire company now called Exact Sciences. And this is just a PGS, which you guys do in your sleep. And that's not even getting into the more advanced stuff. And so Greg, who's a colleague of the Grail founder, took the idea and said, here's what I think of. Let's look at a human life and life itself as a convex hole in tensor space. I'm like, what? Yeah, if you think of tensors and that's a multidimensional space, the viable life is actually a shape in that space. And then we can look at our human life as basically a path through that shape. So all I basically need, he said, is I need to go get multimodal data to figure out what the shape looks like. And he did this by he started in one area, he looked at online Indian exams. So in India, they actually publish your exams and how you did as a way to sort of, vet new doctors. So he gathered these up and basically trained a model where he's able to get an 86% score, which is barely passing, after basically taking a model off the shelf, no fancy trickery, no new changes, no new hyperparameters, just cleaned the data and fed it in a month, pulled the paper together at the last second. He said, we're 86%, let's publish. And I found out about this work when he published it. It was so fast that my customer told me about the research, and I'm supposed to know about this at Google. And that was last December. He's now at over 99%, just continuing to refine the model and refine the data. And what I want to leave you with is now I'm going to give you a tour of where this technique of reducing the different modalities of data into a coding problem are now doing things that we used to read about in science fiction and many of us have dreamed about. Here's one. This is almost biblical in a sense. There's a type of blindness called LCA blindness. And it happens. And the reason I'm poking on LCA blindness is that it's a blindness that happens after you're born. The reason that's important is that your brain is able to process visual signals and see things. So you have a neocortex that can process a signal. And it turns out what they're now doing is that in LCA blindness, they're using CRISPR and looking at your DNA sequence itself, and they can inject a custom sequence into your eye. My uncle actually had this. And what actually happens is it goes in and changes the germline of the DNA in your retina, and it's isolated enough from the rest of your body, and blindness is restored. Now what I mean by that is that people can now start to see shapes and other things before they hadn't seen light before, and now suddenly the lights get turned on. And how much the blindness restores is how much your neocortex is able to process that. So now, basically looking at coding, you can actually recode your retina to actually be able to see. David Sinclair, you might know him, he's on the right. I call him he's like the Tom Cruise of healthcare. Basically, what David Sinclair's been looking at, he has this information theory of aging. He's been trying to get published for about a decade. It finally hit Nature, I think, in January this year, or December of the year. And his idea, which is rather controversial, he said, we're born as a baby. That's not controversial. He said, but then over time, your DNA starts to get damaged. And he said a lot of that's largely from the sun. It could be from drugs, other things. And what ends up happening is your genome gets a little unstable, and you get these cellular senescence. In other words, cells forget who they are. It gives you sort of the cricks and creaks of getting older. And over time, once you lose cellular identity, once you have cellular senescence, that leads to disease and finally death. And his theory is, can you start to understand what happens in this phase and send things back? In other words, if you start to see information degradation, can you start to look at the sirtuins that cause that and correct it, like telomeres, and reverse aging to essentially now basically keep your DNA copy clean? And you can see the paper in Nature. I highly recommend it. It's called The Information Theory of Aging. But now they're starting to look at things about how, very famously about a decade ago, how if you, for example, taking wine, for example, these molecules that are actually used and are surviving in very tough conditions actually activate these sirtuins that actually help you repair the information in your epigenome. Pretty amazing. Again, this is about using programming and a program methodology like Phenomics to understand your body and reverse aging itself. Chris Mason, who worked on the Trent astronaut study, Chris said, well, why are we stopping at humans? I said, what do you mean? He goes, well, look, we have eyeglasses. I wear eyeglasses. We have braces. We have we basically have heart implants and everything else. How about looking around nature, and can we borrow genomic sequences from other parts of nature as ways to assist us? And since he's interested in space, he said, if we're going to Mars, radiation's a problem. How about these little things, these little tardigrades? Tardigrades are these fascinating creatures. They're actually on the moon now. A tardigrade you can desiccate, in other words, take all the water away, and it looks like it's dried up carcass, like it's dead. Leave it for 6 months, add water, they come back to life. Well, how's that happening? So a team in Japan did a genomic analysis of this and did a GWAS basically on these little tiny bugs, and they found this part of the gene they called damage suppressor. Not very inventive, but DSUP is the name of the gene. And so he took the damage suppressor gene, ordered some liver cells with a few clicks on the web, you can do that today, and basically took these and stuck them in a microwave, essentially. And then looked at how long do these cells survive if they're actually radiated. And what he found is that the control group here is basically in red, the vector, and with blue, he's able to actually suppress and have these cells not die as fast by actually having the gene intermix in the germline of the liver cell itself. So the idea here is, could you actually take a pill instead of lathering yourself up with sunscreen, is the idea. And what's now happening is if you look at companies like Ginkgo and Moderna and others, these cells that actually produce the actual vaccines I talk about, it's a robot that looks much like this, and the robot is acting like a lab tech. And they're continuing to make these cells smaller and smaller, much like Amazon does or we do in our data centers. It's so small, soon it's going to fit in a Sprinter van, which you see all over Tahoe. And you can actually make a clean room inside the Sprinter van. What they're now talking about is, could I actually get the manufacture of these drugs into remote areas or outside a city so that I actually can repair someone within 72 hours? And so now they're talking about manufacturing these drugs in a distributed center, which distributed clean rooms as opposed to individual, so you can actually have this across the globe. And to program these things, this is happening now. This on the left, you can see this company called Replit, where it's actually the browser itself as your visual IDE. And as you're programming, part of the programming is a fermentation step. So the programming runs an LLM that actually does actions that control the robot that actually go test to come back in. So now as you're writing code, you're actually controlling robots that are part of your test. So as you compile, edit, and debug, there's a fermentation step as well. And this now becomes a natural way. So they're programming these drugs and everything else inside a browser now. And what you see before is that now they're starting to take raw genomic data, and you can take a sample, and the cost of this used to be $1 billion, it was $100 million, it was a few $1,000s. In the lab today, Andreessen Horowitz invested in it, it's $100 to get a whole genome sequence. With the 100,000 molecules that we actually study, if you do GenDiome, you can do it for $5. And most of that is reagents. So you can go from a sample from raw data all the way through to understanding the care in a car. And this is all happening because of what Lee calls hyperscale AI, because it's all powered by cloud. And the next thing you're going to see is because Moore's law is dead, in order to process these tensors, you need a new type of computer. And so what we're seeing now is on the left is our TPU. You see this thing called a Dragon chip, which comes from Illumina. The idea is if you look at the signals, you want to process the signals at scale. So basically, the Dragon chip is borrowing things from the telco industry, which is all radio signals, and that's an FPGA. And then we're looking at tensors to actually make machines that are just process the tensors themselves. And what's cool about these machines is it's approximate math. Because a lot of the optimization you do is like skiing down a hill, and if you look at the tensor math, you can say, what's 8 times 7? 56. Perfect. What's 6 times 10? 56. Wait, 6 times 10? Yeah, because it's close enough, like horseshoes, as you're going down a hill for optimizing a surface. So the science behind this is mathematics, but basically, if you have sufficient accuracy, you don't need the high precision to do the math, which saves you a lot of energy. And the next thing we're doing is because you want to help people across the planet, the data itself is quite heavy. When you think of nanoscale data, we have multiple internets in a single human body. So you've got to be able to move these data across the planet. And so what we've done at Google is we actually build our own fiber, and we can actually snake in new glass across the tubes. And the idea here is that we need to be able to find ways to move multiple copies of the internet at very quick speed so that as you're programming in the browser, it just feels like it's all local. If you ever play with, when I first worked at Google, I remember sitting down doing some analysis on machine learning, and I had a large data set, several gigabytes. And I hit copy, and I stood up, and it returned. Like, damn it. I sat down, I hit copy, it stood up, and it returned. I said, something's going wrong. And she's like, no, look at the file. I ran a checksum, and the file was accurate. And the reason is because these things are designed at petabit speeds, and that's the kind of speed you need to be able to iterate at the speed we're talking about before. And we're now thinking about is how do you start to make this systematic? So if you have EHR data, imaging data, genomics data, social data, wearable data, you want to have pipelines where you can literally in Python or R or JavaScript be able to take these and turn these into tensor spaces that you can then reason about inside a browser to do the analysis that you've been talking about. And what we're now thinking about inside Google, and Jeff Hinton did an essay about a year ago. He said, what I really want to be able to do is run an LLM in a toaster and have less draw than my toaster. Toaster about 700 watts, by the way. It's about a single GPU. And the idea here is that can we start to look at new computing styles where the computer itself is more focused on thermodynamics and it's disposable. So the idea here is that could you actually have computers that expire like milk? And what we're looking at then is what does that stack look like so that now you can actually program these things on the edge and have LLMs basically at the power inside here. There's some recent stuff you might have seen. December 1st, Lex Friedman had an interview with sort of the EAC crowd. They're now thinking about, is there a new way between quantum and regular computing in the thermodynamic space to understand that? We don't know. George Hotz is looking at this as well, but we're trying to figure out what's the new computing fabric to be able to do this, because in Phenomics, we're reducing all the modalities to basically tensor space reasoning, and that's how we're sort of finding these interesting patterns. And so I sort of want to end on a note that there's a new, like 6 months old now, Lee and I and some others put out an issue with Scientific American that kind of walks you through what's happening in this phenomics space. And I think the basic idea is you can take the different modalities, you can reduce these tensors, and now you are reasoning in this Latin tensor space, and we're finding all kinds of emergent properties from this. And hopefully, as we continue progress, we're going to see in this next decade, we're going to start to see personalized cancer vaccines that attack the thing that I promised my mother many years ago. So thank you very much for your time, and if you have any comments or questions, be around for lunch, but thanks again for your attention.
Hi, obviously your talk was very impressive. Thank you so much for that. And my question is more on the end of the human side of things. I don't doubt the capabilities of technologies and all of this advancement, but do you guys also are taking into consideration how, society will react to like these novel technologies? I'm just thinking also from the point of view of, the pandemic. We were enforced to get vaccines, even people that didn't want to, other ones were not as receptive. makes me think of like, even if we're able to get this machinery like into very rural areas, I've been there and people are not as welcoming. So how would you plan, or how do you think it will go, having this mass advancement in technology and paired with how do we perceive drugs and new advances?
Yeah, that's quite a lot to unpack. A couple things. What we're seeing now, I mean, I hope you saw from the slide, and a lot of this research we're seeing across the globe, is that AI is becoming, it's sort of the newest entrant into the lab. And we're now starting to see AIs moving from a tool to being a true collaborator in science. And I think that's what's interesting to me, is in other words, it's now become a collaborator. NASA, who's very, very risk averse, one of the most risk averse organizations I've ever met, and they look at AI and these tools as someone has a cool idea at lunch, or someone has an idea at the bench, and they take that idea with a big grain of salt, but they use all the classic techniques of science and ethics and other reviews they do for all other science to validate that idea before it goes out, right? I think what you'll see on a lot of magazines is people talk about AI, the AI doctor, the AI diagnostician, the AI making these decisions, AI self-driving cars, for example. What we're finding, though, is that that's much more problematic because of the issues you talked about, right? Letting the AI make the decision. If instead you bring the AI in as a collaborator for the science, we're thinking that can rapidly accelerate our own thinking, and that's really where the better tool is. And all the issues you talked about are like any other technology, right? And all the issues about how do you get, for example, if you want to help out people in Africa, the issue is not getting them the medicine. The issue is they don't have water, right? They don't have a clean way, a good place to sleep, right? Or basic food. And so basically, to summarize what I'm saying is that we look at AI as a collaborator, and that's where we think the success is going to come from. And all the issues you talked about in terms of privacy and ethics and how it impacts humans, it's just like any other technology. And it really comes down to actually a group of humans deciding and working through those issues by themselves, right? The danger sign is when people say, oh, just let the AI go do it. We haven't seen any success there. Question?
Hi, Sam Picos, Institute for Systems Biology.
Oh, great. You know Lee really well then, yeah.
Yeah, he's my postdoctoral advisor. So I was wondering if you could talk more about how we can get stakeholders in the scientific community on board with using AI and break out of like these data silos that we're in, because that's going to be limiting. And also, how do we like retrain and make sure it's accessible to the users that are students and researchers to make sure that this becomes like a helpful tool moving forward for research?
Yeah, I don't know if I have answers for that, but I have a couple suggestions of what we find useful. Scientists, like all of you, healthy dose of skepticism, right? We need to see things validated. We don't see research that we can reproduce. You know, we see an interesting result, like show me the data, show me the process, show me the output, show me the reviews, right? So I think we're going to start to see is more and more of AI being used actually in science. I haven't yet seen where AI is cited in a paper, but I have personal experience where actually writing now with an AI is actually where you're the creative driver and it's more the grad student. That's a really useful use of that. And I think it's more about us starting to use these tools in science and then cite them and reference to them actually and show people how it uses. The second thing is, I've found inside is that people say, well, what's this generative AI thing? What's everything else? There's nothing like actually showing someone how to use this effectively, and then their eyes open up, right? So it's almost like you got to lead them to the water to drink and say, walk me through how you actually use this tool. And then if you take the NASA approach, as they use a tool in science, it's like a microscope, right? And then that's going to allow them to publish much faster than their peers. Like, how'd you get that done so quickly? How'd you go through the experiment so fast? Or like that drug, how'd you find a drug in a day? Because the cost of 6,000 drugs by 150,000 is intractable. The AIs and its savant goes, try these 9, right? And I think as we start to see more and more of the scientific community, I think that's how it's going to be embraced. You really can't just tell a scientist to go use something.
Great, thanks.
That's my guess.
Hi, Rachel Karchin from Johns Hopkins.
Oh, hey, Rachel.
I'm wondering if you have thoughts about interpretable AI, particularly when you talk about clinical applications like, really precision cancer vaccines, but you're also talking about huge tensors going into a system and huge tensors coming out of the system.
Right.
And then you're looking at clinicians who really want to understand why a machine has made a decision. So I wonder if you have thoughts about progress in that direction.
Yeah, a couple things. So what I find interesting about the question, I get that a lot, is that we work with things all day long that we don't know how they work. In the sense that I may work with you, and I don't know what neurons are firing as I ask you questions, but if you think of AI as a collaborator, how do you trust someone's individual work? How do you validate their contribution to a project? And a lot of those techniques, I think, are going to apply to AI. That's one thing. The second one is we're now starting to see where basically LLMs are being used to basically evaluate LLMs, where now it can actually walk through its chain of reasoning, which is it's like reverse engineering the prompt. So that's pretty interesting. We're now, because it's a machine, we expect so much more from machines than we do humans, which is sort of counterintuitive. Like a washing machine, we want it to just wash our clothes, right? And so we have an AI as a scientific. We actually expect more from the AI than we do from the humans, which is interesting. But now we're starting to see, can we start to use an AI and actually train it? So like you have a human that walks through the analysis and a machine, and the machine can say, here's the thought process, and have the machine then reproduce that. And that's really interesting. We do that sort of things at Google where we first do a deep learning, and it works beautifully. Like, that's amazing. But it's nothing like a great feature, right? So we do a lot of research once we take a deep learning output and figure out, is there a simpler model? Sometimes it's if then else. But is there a simpler model to understand that we can actually explain and use that, especially when we deal with money, right? So it's like we use the deep learning to find the first one, but then we iterate to find something we can actually explain. So there's another approach we're looking at. But great question.
Hi, Zach Moss, University of Colorado Boulder. So you seem like you have a very good high-level perspective of a lot of these problems. What do you think are the most pressing unsolved problems in just the field generally right now?
I think that one of the biggest pressing ones is watts per inference. If you look at how much power we're using in these models compared to the human brain, we have like 10 to the 5th, 10 to the 7th to go. And I think we need fundamental breakthroughs in how we compute and how we represent. I think there's interesting approaches in physics-based representations of AI. Quantum may be 10 to 15 years off, but are there new ways to represent this? And so what I'm also finding are there more concise and emergent properties of these models? So for example, there's something called Video Poet that some researchers just produced. I was probably surprised as many of you were. If you look under the covers, they represent frames in a video as a 2 to the 18th bit or an 18-bit integer. And that works, which is astonishing, right? Because imagine all these models you had for years of understanding what's in an image, you can't reduce it. Well, apparently you can if you're trying to understand videos to actually a single frame. So these are the kind of step forwards we need to see, which is, are there new representations? Are there new ways where you got to get the watts per inference down? So we're now about, like a single GPU is like 700 watts, right? That's like a little oven. You need 4 of those, right? 8 of those on the big one. So 5,600 watts, right? Versus a human, if your head hurts, you're burning 25, right? And you're much better than AI. So we've got a long way to go. Right? So that's a fundamental problem. I think that's new computing, new representations, new ways, and then looking at emergent properties of transformers. Very cool stuff. Question?
Scott, that was a lovely high-level overview, and I agree with you about the watts per inference thing, but I want to go in a slightly different direction.
Sure.
And so, Google likes to say that they're organizing the world's data, and these models depend crucially on the data that's being used to feed them.
Right.
And so I have kind of 2 questions for you. One is, what data do we need most? And I'll just point to one of your competitors. So Meta just announced this thing where they're following people around and doing head-mounted cameras and around cameras and all kinds of sensors to try and capture the daily life of what a person walking around in the world looks like to try and make predictions.
Yep.
And so that sounds like an interesting kind of source of data to train on, and I'm kind of wondering what you guys are up to with respect to what are the exciting new sources of data we want to train these kind of models on. Related question is, using other people's data to train your models is getting some of your competitors into trouble. OpenAI just being sued by the New York Times.
Yep.
Lots of memorization and things. If you prompt the models correctly, you can pull out copyrighted materials and the like. And from my perspective as a scientist working in this area, the hard part is not, did you pay for it or not? I figure you guys can work that out among yourselves. But how are we going to know what training data was used? Because it's really critical in understanding the performance of these models to understand what they were trained with. For example, to tell if something was memorized or not. So could you talk a little bit about what kind of data we need and what kind of openness we need?
So I'm a student of Lee's, and he's on this path, and he has been for a number of years, especially in medicine. He calls it the million patient problem. He said, if you just gave me, he says, 1 million patients over 10 years in the phenomics, I will completely change healthcare. I said, what do you mean by that? He said, healthcare is sick care today. You go in because you feel sick, and then we treat the sickness, as opposed to going in and looking at a phenomic correlation that could say, you look like to be precancerous. Here's a few things to steer your gut biome to protect that, right? So it shifts to much more wellness. Now, the problem with that is one phenomic sample in 2016 was $6,000. So he said, I just need 50 billion to do the first million of those. So that's where we're at today, is like, how do you go get 1 million samples of phenomic data over 10 years? And that longitudinal data will be a wonderful thing for science. That does not exist today. Now that data requires everything from the watch you might wear to the Oura ring to different scans. That's a data set I think that will actually help us change medicine. I don't know how we're going to get that yet. Now the second thing is in terms of where the data comes from. We're starting to see much more in the AI community. We do this internally as well. Whenever we actually use a model in production, we actually take that as well as the data, and the data set itself is then correlated with the model and the release version and the weights, and that's actually time-stamped, and we use that to reference a model, so we can always go recreate if there are error conditions. Now, in terms of actually attributing back to where it came from and what original source, that's an area of wide open research. Because essentially, these models are basically predicting sort of the next token and they're all hallucinating, right? Except some of those hallucinations will line up with what we perceive as being truth, and others line up with things we don't believe to be true. And so what we want to do is, and what we're now finding is that LMs are great as a control plane, but if you want to do a source of truth, you need much more like a search engine itself to go find the source of truth. And then it's a matter of, well, that's true according to what? So for example, in religion, there's different religions' beliefs in terms of what might be truth. The same thing happens across other areas. So basically in summary, what I'm saying is there's an awful lot of multimodal data we want to accumulate in a longitudinal way to healthcare, and we're starting to see different companies have pockets of this, and I'd like to see that come together in nation states. And the former is, we now not just looking at LLMs, but LLMs with things like information retrieval to go after the source of truth and where it came from. And these are steps towards that. But these are, as you're implying, these are wide open research problems.
Hi, Julie Lynch, Department of Veterans Affairs. Your comment about the million patient data was very interesting because one of the best taxpayer-funded resources is the Million Veteran Program. Mm-hmm. And I think that that doesn't solve many problems. I mean, as somebody who's been, deep into the 20-year longitudinal electronic health record data on these million veterans, you have underdiagnosis, misdiagnosis, underrepresentation of minority populations right. in these million patients. And so, we've been trying to do a machine learning algorithm for hereditary amyloidosis, which is, an underdiagnosed, misdiagnosed disease, and it's extremely challenging. And so I just wanted to point out that the million patient data does exist within the Department of Veterans Affairs, and even with all of those data, there are still many challenges.
Yeah, well said. Now, I think if you look at, there are a number of VHRs, for example, you see this more, certainly in Israel, we're seeing this in Singapore, we're now starting to see the UK. We're now starting to bring in, along with the EHR, can you bring in other phenotypical data as well as genomic data? And that's sort of interesting. But I'd love to talk more about what you found and what we're looking for. Last question?
Yeah. My name is Kwesi Ebuafihene, and I'm from Howard University. Essentially, I want to solicit your thought. Currently, the debate around AI kind of have two issues. Like, some say it could be a great asset to society, and some say it could be a menace to society. Mm-hmm. What do you think is the best way society can manage it in a way to keep it as a great asset? Do you think it's ethics or regulations? Thank you.
Ethics or what's the second thing?
Regulations.
Ethics or regulation? That's an interesting question. I think what we're seeing is you all are now starting to see what we've seen in the labs for a number of years with ChatGPT and the release of these LLMs. And this is a brand new, basically a probabilistic model to help us understand and automate something we thought would not be the first thing to automate. We all thought it would be robotics initially and blue-collar work and mechanistic work, much like we did with farming. And that's how we got a big agroeconomy, because we had these farming implements. So the natural thing was we thought it was going to be with robotics and everything else to do that labor. And it turns out that we can actually iterate much faster in the cognitive realm. And so now we have these new emergent models. I think what we're seeing is, there's an interesting point where they talk about how powerful these are today, and we're saying, well, they're almost superhuman. Well, yes, they are. But we also have other superhuman tools, too. For example, a hammer is a superhuman thumb. A drill is a superhuman screwdriver. And we use these all day long to continue in how we produce them around our own globe. And now we're trying to figure out is, with these new tools, there's a lot of talk about AGI, for example. And an AGI becomes much more about, it's almost like Earth-centric. We talk about that in terms of a milestone, in terms of, is it generally intelligent as us? And I look at it as AGI as much more about, is there an algorithm that's general purpose that can operate with sparse data and do something interesting that's human level? And we're getting very close to that. But now we're starting to see is that is a one stop along to what's the real definition of intelligence, and that's really exciting for us. And we have some tool like this, much like we had with drill bits and the rock and gunpowder and other things in the past. How do we then come together as a community and realize where is appropriate use of this and where is not? And I think that has always been a collaboration between science, government, and society. And those sort of standard mechanisms, I think, are going to be needed brought here. But this is basically, it's a progress of human ingenuity, where we're now on the cusp of actually understanding the neocortex as a tool. And as Nanoscale says at MIT on a Lex Freedom podcast, AI is not a tool, AI is much more a collaborator, which is an interesting thought. And so as you bring that in, the same we've done with all these powerful technologies in the past, we'll sort of iterate through those, and I think it's a combination of technologies and regulation to get us through this successfully.
Hi, great talk. Jeff Rhodes from Genelia Research Campus. So you mentioned a bit about the different computing structures using TPUs, et cetera, and you use this analogy of the neocortex or the brain to sort of understand or imagine how this tensor processing happening. But in the brain, there is a lot of heterogeneity in how compute's happening, right? It's not just a simple tensors in and out and like t equals 1, t equals plus 1.
Yep.
So do you foresee any sort of heterogeneity in compute infrastructures as well to sort of incorporate or mirror the heterogeneity that actually exists in the brain?
I love your insight. If you think of Moore's law, the idea is you take the same circuit and you draw it again half as small, and you get magically you get more compute power every 18 months. That's largely dead. What we're now seeing is that the way you're getting more performance is through heterogeneity, where you look across those pipelines, and I think as you're figuring this out, what are the different steps in computational structures you need to pull these together? And you saw some emergent ones today I walked you through. For example, the element itself is a fantastic technology, so is the transformer. This notion of using tokens, which is the video poet I just saw, that's fascinating, right? It's an emergent property. But that actually can be used in the good old-fashioned AI as a tool, and these are starting to come together. And if you look at big areas for research, LM it's very short of memory. It needs long-term memory, right? It needs the ability to interact with the outside world, right? And that's more the idea of adding the ability to tools, right? And then those tools, I think, are going to come together, and the answer will be, I think you nailed it, is it's not going to be all in a von Neumann box, right? It's much more a heterogeneous leveraging of different technologies that pulls off what you and I would consider an intelligent partner, right? And right now we've seen a fascinating with LLMs. That became a fascinating with APIs, but now we're seeing mixture of experts is really where it's headed. So I think your intuition's right on the money. But the answers, you're all going to create the answer. I don't know what the answer is.
So I want to ask a question related to the question before last.
Sure.
And that is kind of, I think it's I think the correct answer, or at least I agree with an answer that, new technologies always need societal, government, right, stakeholder interest in figuring out how to regulate them. But in the current world, FDA and its international peers are basically the most rational, data-driven people in any regulatory environment, right? FAA is pretty good, data-driven. People in transportation are pretty, but FDA actually has a real formal approach to regulatory science, and the people actually research how to do regulation. Do you have thoughts, and I don't know if anyone here is from FDA, but do you have thoughts around people who are working in AI in biomedicine to kind of take a leadership position generally and work with regulators to say, this is how we will regulate and demonstrate a data-driven approach that actually has a kind of rational framework, as opposed to, like, there's Biden had an executive order that was actually taking power away from FDA and putting in groups that don't know how to do regulation. So do you have thoughts about how people in biomedicine can work with regulators to actually provide prototypes for how regulation may proceed?
Good question. I think what I'm seeing is, the companies I've worked with, and we actually work with some people from the FDA as well. There's actually a lot of brilliant staff within the FDA. And what I like about what they do is they bring scientific rigor to a lot of these regulatory approvals, right? And so what we're now starting to see is emerging conversations are, can regulations now start to be code as well as just as English, right? And so that's allowing to sort of an additional level of rigor in compliance, for example. And I think that's sort of an interesting area to explore, where now leading companies I've seen in biosciences and mRNA-based drugs and everything else are now actually talking to the FDA about what does a platform look like? How do you approve that process? How do you approve, and is it really going to be with paper, or is there other ways to use code as well for that? That's sort of an emergent idea. But it is a thing where it is really a true collaboration between the scientists at FDA, the scientists in industry, trying to find what can we do, and what's the least amount of regulation we need, but no less, to actually make this safe for people, right? And that's an open question. But I do like is that we are seeing, and we're seeing it in the White House and everyone now starting to convene scientists with industry, with the regulators, and have those conversations. And I've been very encouraged with what I've seen personally in those conversations.
I think we're good, yeah. Without any other questions, let's give Scott another round of applause.